Sure. So TLS or tertiary lymphoid structures are conglomerates of T and B cells that are found in the tumor microenvironment and has been described to be associated with favorable prognosis in different cancer types, including bladder cancer, which we had contributed to in the past, looking at a number of muscle-invasive bladder cancer patients from whom the presence of tertiary lymphoid structures surrogated by an increase in the 12CK score have predicted not only better prognosis in patients who are treated with surgery alone, but also in those patients who are with metastatic disease who are treated with immune checkpoint blockade. And in line with that, there's also been several different studies looking at the response to immune checkpoint blockade being predicted to be higher in the context of tumors that exhibit high density of tertiary lymphoid structures. So in this study, we really took it a step further and studied the density of tertiary lymphoid structures both before and after the combination treatment with the intravesical oncolytic virus, as well as the immune checkpoint blockade. And what we found was that even though at baseline, the density of TLSs did not predict for complete response, but there is an induction of tertiary lymphoid structures in the post-treatment samples that were only found in the complete responders. And not only that, we also found that there is an increasing density and increasing maturity of these tertiary lymphoid structures, as evidenced by an increase of CD138 positive plasma cells that exist both within the tertiary lymphoid structure and also found in the surrounding stroma, suggesting that the B cells are undergoing this evolution from a more naive state to the more mature state of CD138 positive plasma cells that then are being infiltrated into the tumor microenvironment to induce the antibody projected response that they are capable of projecting. And not only that, we also found that there was evidence of isotype switching, whereas at baseline in these pathologic complete responders, we're more likely to find IgM positive tertiary lymphoid structures, that after the treatment, these tertiary lymphoid structures really switch their isotypes to either an IgA or an IgG response. And further corroborating the antibody-directed response, there was one single patient in our population who had a transient partial response at the time of the tumor biopsy after treatment, who then went on to become a complete responder. And from this partial response specimen, where there is this transient level of residual tumor cells, we were actually able to detect the binding of IgG on top of the remaining tumor cells. Again, highly suggestive that there is an antibody-directed response against the tumor cells themselves.